The overall goal of this procedure is to compare two key functional characteristics between therapeutic antibodies, target binding and CDC induction, during their biosimilar development or as a quality control during their production.
These methods can help answer key questions about how to assess functional differences between therapeutic antibodies that bind the same target protein. These techniques are quick, simple, and inexpensive, allowing for their execution in most laboratories. Demonstrating the procedures will be Edith González-González, a research associate from my laboratory.
Begin by transferring the DOUDI and DOUDI GFP-positive cell suspensions into individual 15 milliliter conical tubes for their collection by centrifugation. Resuspend the pellets in 5 milliliters of PBS for a second centrifugation, followed by resuspension in enough PBS for counting. If the cultures are 95% viable or greater, dilute the cell suspensions to a 4 × 10⁶ cells per milliliter of cold staining buffer concentration, and aliquot 50 microliters of cells into individual 1.5 milliliter microcentrifuge tubes containing 100 microliters of at least 5 different concentrations of the reference or biosimilar monoclonal antibodies. When all of the cells have been transferred, incubate the samples at 4 degrees Celsius for 20 to 30 minutes, followed by centrifugation in 1 milliliter of PBS per tube. Resuspend the pellets in 100 microliters.